So now we can put together the whole life cycle of the angiosperm, and you can see that this is still alternation of generations. If I draw my line this way, this side is my sporophyte generation, and this is diploid. Everything on the blue side is my haploid gametophyte. But this one's been filled in with all of the details for a flowering plant, the angiosperms. So we'll start here again with our diploid sporophyte adult flower. It's got two sporophylls. We have the microsporophyll, which is the male reproductive organ, which makes microspores. And then as they go through meiosis to become a microspore, now this is haploid. We also have our female sporophyll, which is the carpel. Inside of the ovary, each ovule is going to undergo meiosis to make a megaspore. And you can see that when we do meiosis, we result with 4 cells. Only one of those is going to become the megaspore. So while this is occurring, the microspore is going to divide. Remember, this division here is mitosis, into 2 cells. The tube cell that's going to make the pollen tube, and the generative cell that's going to eventually be 2 sperm cells. Pollination is happening here. So here's pollination, where we have the pollen grains landing on the stigma, and that tube cell is going to lead the way. And as the generative cell follows behind it, it's going to again do mitosis to become 2 sperm cells. And then this is going to travel all the way down to the ovule. And it's following the signals sent by the synergids, which tell the pollen tube, hey, come to my micropyle, I've got an egg for you. By the time the pollen tube reaches the micropyle, this will have developed into the female gametophyte, which is the embryo sac. Remember, this embryo sac has these 8 cells. There's 3 antipodal cells, 2 central cells, 2 synergids, and 1 egg. So if you add that up, it should be 8. And then we have fertilization, but not just one time. We have double fertilization. One of the sperm is going to fertilize the egg, which is going to develop into a diploid zygote. This is the embryo. The other sperm is going to fertilize that polar nuclei, which is going to develop into the endosperm, and this is triploid, it's 3N. And that's going to eventually become the food source for the developing embryo. And this embryo is going to develop inside of my seed. My seed is made up of 3 components: an embryo, and an endosperm, and a seed coat. This seed coat, the 2N from the seed coat, comes from this plant. It's from the parent plant. The embryo and the endosperm are results of fertilization. The seed coat are the integuments from the original ovary. Eventually, as the seed develops into a mature seed, it'll find an environment that it is going to germinate in, and germinate into a seedling, and then eventually grow into an adult plant that has reproductive flowers of its own.